Welcome to the Eukaryotic Microbial Atlas. My name is Professor Triwibowo Uono from the Department of Agricultural Microbiology at Universitas Gadjah Mada. In this presentation, we will embark on a journey to explore the fascinating world of eukaryotic microbiologists. Our journey begins by understanding the fundamental principle that defines eukaryotic cells: compartmentalization. Imagine a cell growing larger. Its volume increases much faster than its surface area. This presents a significant challenge for nutrient uptake and waste removal, as simple diffusion becomes inefficient. Eukaryotic cells have ingeniously solved this scale problem by folding their membranes inward, creating specialized internal compartments known as organelles. This not only dramatically increases the internal surface area, but also allows for incompatible biochemical processes to occur simultaneously within the same cell, leading to greater efficiency and complexity. At the heart of the eukaryotic cell lies the nucleus, often referred to as the cell's information vault. It's enveloped by a double-layered nuclear envelope consisting of two lipid bilayers separated by a paranuclear space. This barrier is punctuated by nuclear pores, which act as selective gatekeepers, regulating the passage of RNA and regulatory proteins. Inside, we find chromatin, where linear DNA is intricately wound around histone proteins, offering protection from enzymatic degradation and oxidative damage. A dense, non-membrane-bound subcompartment within the nucleus, the nucleolus, is dedicated to the crucial task of ribosome assembly. Moving beyond the nucleus, we encounter the endoplasmic reticulum, the cell's biosynthetic supply chain. It exists in two forms: the rough RER, characterized by its studded appearance due to ADS ribosomes, is the primary site for the production of proteins destined for membranes or secretion. Here, proteins undergo folding and glycosylation. In contrast, the smooth SER, a network of tubules devoid of ribosomes, is involved in crucial processes such as lipid and steroid synthesis, energy storage, and detoxification. Proteins and lipids from the ER then move to the Golgi apparatus, the cell's logistics and routing center. The Golgi functions by reading carbohydrate shipping labels, such as mannose 6-phosphate, to direct molecules to their correct destinations. There are several potential paths. Some molecules return to the cytosol if mistagged or are ER-resident proteins. Others are directed to the cell membrane to become protein channels or identifiers. Some are packaged for secretion via exocytosis upon receiving a chemical signal. And finally, some are sent to lysosomes, delivering hydrolytic enzymes. Maintaining cellular integrity and recycling cellular components are the roles of lysosomes and peroxisomes. Lysosomes are essentially the cell's recycling centers, filled with potent hydrolytic enzymes. Their internal environment is highly acidic, with a pH of 5, compared to the cytosol's pH of 7. This pH difference is a crucial safety mechanism, ensuring that these enzymes are only active within the lysosome and do not damage other cellular components if accidentally released. Peroxisomes, on the other hand, act as the cell's security system, primarily involved in fatty acid breakdown and neutralizing harmful reactive oxygen species, such as peroxides. The energy powerhouses of the eukaryotic cell are the mitochondria, and in photosynthetic organisms, chloroplasts. Mitochondria generate ATP through a proton gradient created across their inner and outer membranes, while chloroplasts convert light energy into chemical energy. Their unique characteristics, such as double membranes, their own circular DNA, 70S ribosomes, and self-replication, provide compelling evidence for the endosymbiotic theory. This theory posits that proto-eukaryotes engulfed aerobic bacteria, leading to a permanent symbiotic relationship that gave rise to these essential organelles, providing structural support, facilitating movement, and organizing cellular components is the cytoskeleton, a dynamic network of protein filaments. It comprises three main types: microtubules, which are 25 nanometers in diameter and made of tubulin, act as rigid tracks for intracellular transport, form the core of cilia and flagella, and are crucial for the mitotic spindle. Intermediate filaments, around 10 nanometers, like keratin, provide stable tension-bearing support, anchor organelles, and support the nuclear envelope. Finally, microfilaments, the thinnest at 7 nanometers and composed of actin, are responsible for cell movement, cytokinesis, and muscle contraction. Now that we've explored the general architecture of eukaryotic cells, let's delve into the diverse eukaryotic microbial spectrum. This vast group includes algae, which are chlorophyll-bearing photosynthetic organisms, ranging from unicellular to macroscopic forms. Fungi are achlorophyllous heterotrophs, exhibiting varied morphologies such as molds, yeasts, and mushrooms. Protozoa are unicellular, wall-less, motile heterotrophs, known for their complex locomotion. And finally, slime molds represent a fascinating taxonomic bridge, displaying amoeboid vegetative states alongside fungi-like reproductive bodies. Our first major clade focuses on the photosynthetic architects, primarily encompassing various forms of algae. These organisms exhibit a wide range of morphology and scale, from microscopic 5-10 micrometer unicellular forms to macroscopic structures reaching up to 3 meters. They reproduce through dimorphic means, utilizing both asexual reproduction via unicellular spores and sexual reproduction through the conjugation of gametes. Next, we move to Clade 2, Kingdom Fungi, a diverse group with significant ecological and economic importance. Fungi can be broadly categorized into molds, which are filamentous fungi whose basic unit is a hypha, aggregating into a mycelium. Yeasts are unicellular fungi that reproduce either through asymmetrical budding or symmetrical fission. And finally, mushrooms represent the macroscopic filamentous fungi, characterized by their specialized fruiting bodies that produce basidiospores. Let's take a closer look at filamentous fungi, often described as spore factories. Their reproductive structures are quite intricate. We can observe the conidiophore, which is the stalk-like structure, bearing phyalides, which are flask-shaped cells that produce conidia or phylospores. This slide also showcases the morphological diversity across genera, with examples such as Penicillium chrysogenum and Aspergillus versicolor, demonstrating the varied forms these spore-producing structures can take. Unicellular fungi, or yeasts, exhibit fascinating reproductive divergence. On one hand, we have budding yeast, exemplified by Saccharomyces cerevisiae, which undergoes asymmetrical division, forming a distinct daughter bud. On the other hand, fission yeast, such as Schizosaccharomyces pombe, demonstrates symmetrical division through binary fission, generating identical daughter cells. These distinct mechanisms highlight the evolutionary adaptations for reproduction within this group. Beyond the microscopic, we encounter macroscopic fungi, most notably represented by the fruiting body of mushrooms. This slide details the anatomy of a typical mushroom, including the cap, pileus, scales, gills, lamellae, ring, annulus, stipe, stalk, stem, and volva. Furthermore, mushrooms offer a rich nutritional profile, providing essential vitamins like thiamine, riboflavin, niacin, B6, and folate, as well as minerals such as potassium, phosphorus, magnesium, zinc, copper, and manganese. They also contain key compounds like betaine, choline, and beta-carotene. Fungi are not always benign. Some engage in biochemical warfare through the production of potent toxins. A prime example is Amanita phalloides, commonly known as the death cap mushroom. Its deadly secret lies in alpha-amanitin, a toxin that strongly and specifically inactivates RNA polymerase II, effectively halting mRNA synthesis and leading to cell death. The lethality is stark. A single cap can be fatal. In contrast, Amanita muscaria, the fly agaric, utilizes a different chemical arsenal, including psychoactive ipotenic acid and muscimol, demonstrating the diverse molecular pathology of fungal toxins. Fungi also form crucial symbiotic systems, particularly with plants. Mycorrhizal root architectures represent a mutualistic association between vascular plants and fungi. We distinguish between two main types: ectomycorrhiza, characterized by extracellular integration, where hyphae weave between root cells, forming structures like the Hardig net and mantle, but without penetrating the cell membrane. In contrast, endomycorrhiza, specifically arbuscular mycorrhizal fungi, AMF, involves intracellular integration, where hyphae penetrate plant cell walls to form branching structures called arbuscules, facilitating direct nutrient exchange. Another remarkable symbiotic system is found in lichens, which are often described as a fungal-algal mutualistic engine. In this partnership, the fungus acts as the structural anchor, providing a physical habitat, absorbing and delivering inorganic nutrients from the substrate, and creating a microscopic barrier to prevent algal desiccation. The algae, in turn, serve as the photosynthetic factory, producing and exporting synthesized organic compounds, primarily carbohydrates and oxygen, to its fungal partner. This exchange creates a self-sustaining system where both organisms thrive. Moving to clade 3, the protozoa, we categorize these organisms based on their diverse modes of locomotion. Protozoa are unicellular, wall-less, achlorophyllous heterotrophs that obtain nutrition by consuming other organisms or organic particles and are structurally larger than bacteria. We have amoeboids, which move via pseudopodia, or false feet. Ciliates utilize vast arrays of sweeping cilia for movement. Flagellates propel themselves with whip-like flagella. Finally, sporozoa are unique in being non-motile, relying entirely on host mechanisms for movement and replication, often exhibiting strictly parasitic life cycles. Our final clade in this spectrum is clade 4, slime molds, often referred to as the amoebozoa enigma due to their fascinating life cycles. They exhibit a common duality, a vegetative phase that is amoeba-like in structure, and a reproductive phase that forms fungal-like fruiting bodies to generate spores. We differentiate between cellular slime molds, whose vegetative state consists of distinct, independent single cells, amoebae, with Dictyostelium discoideum being a key model organism. In contrast, acellular slime molds have a vegetative state that is a massive, single multinucleated mass of protoplasm called a plasmodium, lacking distinct cell boundaries or a definite size limit. To further appreciate eukaryotic diversity, let's engage in comparative cytology, starting with macroeukaryotic architecture. When comparing animal and plant cell models, key differences emerge. The animal cell is defined by the lack of a rigid cell wall, the presence of lysosomes, and a highly centralized nucleus. In contrast, the plant cell possesses a rigid outer cell wall, photosynthetic chloroplasts, and a massive central vacuole, crucial for structural turgor. These evolutionary hardware differences reflect their distinct ecological roles and adaptations. Extending our comparative cytology to microeukaryotic architecture, we observe distinct features across different microbial groups. The algal cell model integrates a motility flagellum, highly efficient chloroplasts, a starch vacuole for energy storage, and a rigid cell wall. The fungal yeast cell model is characterized by a distinct polar butt scar, evidence of asymmetrical reproduction, prominent storage vacuoles, and a heavy structural cell wall. Lastly, the protozoan cell model stands out with the total absence of a cell wall, replaced by a flexible pellicle, distinct water vacuoles for osmotic control, and complex external glycocalyc structures. Finally, let's discuss the methodology, the microbial identification framework. This comprehensive approach involves 6 key areas. First, culture characteristics, which assesses specific nutrient demands and defines the physical environmental limits for survival. Second, morphology, involving microscopic analysis of overall cell size, physical arrangement, specific differentiation, and internal organelle architecture. Third, metabolic profile, mapping the unique biochemical pathways and enzymatic reactions central to the microbe's life cycle. Fourth, chemical composition, determining the specific molecular makeup of cellular components, such as detecting cell wall chitin versus cellulose. Fifth, antigenic properties, evaluating immune system reactivity and profiling distinct surface markers. And finally, the gold standard, genetic analysis, which utilizes ribosomal DNA sequencing, specifically 18S rDNA, for classifying eukaryotes, whereas 16S rDNA is reserved strictly for prokaryotes. That concludes our in-depth exploration of the Eukaryotic Microbial Atlas. I hope this lecture has provided you with a clearer understanding of the complexity, diversity, and fundamental importance of eukaryotic microbes. Thank you for your attention, and I look forward to seeing you in the next class.